This lesson provides an overview of meiosis, and here are your objectives. First, let's go back to mitosis. Mitosis produces body cells, or somatic cells. So all the cells in your body except for gametes, sperm and egg cells. And somatic cells, or body cells, are diploid, or 2N. They contain two full sets of chromosomes, one from your mom, one from your dad. In contrast, meiosis, which we're talking about in this lesson, produces gametes, the sperm and egg cells. And gametes have to be haploid, or 1N. They contain only one complete set of chromosomes, because at fertilization, a sperm and an egg cell will join together, and that will recreate the diploid state. So in order to have genetic information from a mother and a father passed to the offspring without doubling the number of chromosomes, we need to have that first. So one of the key functions of meiosis is to have the number of chromosomes. And again, that's in preparation for fertilization. Another function is to increase the genetic variability of offspring, whereas mitosis produces two genetically identical daughter cells, meiosis produces 4 genetically distinct or different daughter cells. And that, in conjunction with fertilization, is going to increase the genetic variability of the offspring. Let's look at the process of making sperm or egg cells by meiosis in an organism for which 2N is equal to 2. The diploid number is 2, meaning when it has a full set of chromosomes, it has just one from mom and one from dad. And I'm going to draw those in red and blue here. Now, here we see that each chromosome has 2 sister chromatids. DNA replication happened during interphase, and so now we have only 2 total chromosomes, but each of them is made up of 2 sister chromatids, and the sister chromatids, these 2 blue ones, are identical to each other, these 2 red ones are identical to each other. This precursor cell is diploid because it has 2 full sets of chromosomes, one from mom and one from dad. In this case, the set is just 1 chromosome, but we still see 2 sets, so it's diploid. In the first part of meiosis, called meiosis 1, that's going to be reduced so that we are in a haploid state. And that happens because the chromosomes from the homologous pairs are going to separate. We're just looking at one pair of homologous chromosomes here, so they separate. And then in meiosis 2, each individual chromosome is going to separate at the centromere into the 2 sister chromatids. And so we are staying in the haploid state. So meiosis 1 changes the ploidy from 2N to N. We go from diploid to haploid, and it's therefore sometimes called a reduction division, whereas meiosis 2 takes us from haploid with 2 sister chromatids to haploid with 1 sister chromatid. We are still just going from haploid to haploid. Now the structures that line up and separate here in meiosis 1, this is meiosis 1 shown in the first step, are the homologous chromosomes. Whereas the structures that separated in meiosis 2, and they didn't really line up, I guess they were attached, so this part is maybe not entirely correct, but in meiosis 2 what separated were the sister chromatids. And that's more like what happens in mitosis. So meiosis 2 is very similar to mitosis. We'll see a little difference in a moment, whereas meiosis 1 has pairing of homologous chromosomes, and that's very different from mitosis. Now I should mention that both meiosis 1 and meiosis 2 have the same order of stages as we saw in mitosis, namely prophase, then metaphase, then anaphase, and then telophase. And you can look those up in any textbook and you can see the specifics of what happened with them. The key to understanding the difference between meiosis 1, meiosis 2, and mitosis is looking at what happens in metaphase and anaphase, namely the structures that are separating in metaphase to anaphase. So let's look at that a little bit further now. Let's broaden this to look at a cell for which 2N is equal to 4. In mitosis, I'm going to draw some chromosomes as long and some as short. So let's say this is from one parent. I've got the long and the short chromosome, and they've been replicated in interphase, so they are 2 chromatids. And then I've also got the long and the short from the other parent. So this is what you'd see at mitosis. You'd see all 4 of these chromosomes, remember 2N is equal to 4, all 4 of these chromosomes lining up at the metaphase plate. And that's what would be happening in replicating all the body cells. In meiosis 1 and meiosis 2, however, when this organism was making sperm or egg cells, you'd see a different story. So again, in meiosis 1, we would get the homologous chromosomes to line up. So we're going to get the red and the blue long chromosomes to line up, and we're going to get the red and the blue short chromosomes to line up. Now notice that there's different ways I could have drawn this. I've drawn the red and the blue chromosomes on this side like this, but I could equally well have drawn, let's say, the red and the red on this side and the blue on the other. And the way that each homologous pair lines up, let's call this homologous pair 1 and this homologous pair 2, is completely independent of all of the others. And we call that independent assortment. And that leads to a lot more variability in the possible gametes, because you're essentially flipping a coin at each orientation of these. And we'll see that in a moment, as these get pulled to one side and these get pulled to the other, they're going to end up in different gametes. So independent assortment, which happens in meiosis 1, is a key factor in increasing genetic variability of the gametes. So now if I follow that through into meiosis 2, I've got the long red chromosome here and the short blue on this one, and I've got the long blue and the short red in this one. And in meiosis 2, we see separation of the sister chromatids. So they're going to effectively separate down there. And so we end up with, I'll just draw these down here, we end up with long red over here, and short red over here, and we end up with short blue over here and long blue over here. Now notice that over the course of meiosis, we've gone from, this was effectively the cell we started off with, it's diploid. We've got long chromosomes and short chromosomes, and we've got 2 copies of each. Now in each of the gametes shown down here, we have the haploid state. We've only got 1 long chromosome and 1 short chromosome. So we've gone from diploid to haploid. Now I said that we were going to end up with 4 distinct haploid gametes, and right now they're not all distinct because these 2 are the same and these 2 are the same. But that's because we've overlooked something here, and that's crossing over, which we'll talk about in a moment. But first, just to sum up, the first thing that increases genetic variability of gametes is independent assortment of the homologous chromosomes, and that happens in meiosis 1. And if you work out the math, because there are 2 options, maternal and paternal, for every single chromosome, you've got 2 to the power of the number of pairs of possible gametes from each parent. So remember that humans have 23 pairs of chromosomes, 22 autosomes, and 2 sex chromosomes, the X and the Y. And so meiosis in humans, in one human, could produce 2 to the 23 possible different gametes. So one individual could produce 2 to the power of 23, 2 multiplied by itself 23 times, genetically different sperm or egg cells. Consider that that's happening in the production of both sperm and egg cells, and one mother and one father could therefore produce 2 to the 23 times 2 to the 23 genetically different offspring, if nothing else were further increasing genetic variability. But there is something that's further increasing genetic variability, and that's crossing over. And that occurs during prophase 1 of meiosis. So let's take a look at that now. For simplicity, let's go back to an organism for which 2n is equal to 2, and I'm going to draw the chromosomes as if they were condensed, although in prophase 1, when crossing over is happening, they're not condensed yet. Remember that these are homologous chromosomes, so they have all the same genes, but not necessarily the same alleles. And so what's going to happen, and this can happen in multiple regions at once, is that we're going to see the non-sister chromatids actually connect. Non-sister chromatids being the chromatids from the opposite homologous chromosome. So here, in this region, we see that they are crossing over, and this cross-shaped area here that is formed by crossing over is called a chiasma. The plural is chiasmata. And the 4 chromatids together from the 2 homologous chromosomes are called either a tetrad or a bivalent. Those mean the same thing. Now, I've also drawn the sister chromatids like this separated from each other, far away from each other, but in reality they're actually held together, so it looks more like this. There's actually a protein structure holding them together, and therefore the crossing over that's happening, because the crossing over is holding the non-sister chromatids together and the protein structures are holding the sister chromatids together, the whole thing is connected. And that becomes important to ensure that you only get one of the homologous chromosomes to each side in meiosis 1. So the chromosomes have swapped a piece of themselves. So this chromatid ends up being part maternal and part paternal, and the same on the other side. Now, because the genes are the same on these two chromosomes, we've still got all the correct genes up and down here, but we've got a swapping of alleles. And so now we've got a combination of alleles on this chromatid that didn't exist before. And so that new combination of alleles on one chromosome further increases genetic variability. And if we go back here now, you can see that if crossing over had happened, which it always does, then for example, there would be a little blue piece there maybe and a little red piece there. And so we would see maybe some red there and some blue on this one, and now these two are genetically distinct, and we would of course have had the same thing over here. So we've got some red there and we've got some blue here, and now we've got 4 genetically distinct gametes. And so the two things in summary that increase genetic variability in the gametes are independent assortment of the homologous chromosomes in meiosis 1 and crossing over, which has actually happened back in prophase 1. Add to that the randomness of fertilization, which egg and sperm create a zygote, and you can see why there is an enormous amount of genetic variability.